throughout the talks yesterday, and I thought, okay, this is sort of framing on my part, because now I'm taking a known toxicant and I'm sort of implying, maybe indirectly, that there are some similarities between the toxicity of asbestos and carbon nanotubes, when there very well may not be. Okay, and so then I was thinking, okay, well, if I say this, then other people are going to use this framing to say, okay, we all know asbestos is bad and it's banned and it causes toxic effects, and now carbon nanotubes do the same thing. So I think as bench scientists, we have a responsibility in how we communicate our results, and how this all plays into the whole risk communication process. I'm not sure, but I do think there's a responsibility on our end there as well. So what we usually get asked as toxicologists are which compounds, or in this case nanoparticles, are toxic and which are not, and if so, which inherent properties govern their toxicity. And the answers to those questions are never clear, based on the complexity that I've talked about. Secondly, another challenge we have is that not all scientists agree. So this is true for all disciplines, but this creates this ambiguity that we've been talking about as well. So I think for a toxicologist, both complexity and ambiguity need to be better communicated across disciplines, and that would help in the risk communication process. So what I'm going to do now is just talk a little bit about some of the data we've been collecting at University of South Carolina. We have a nano center there that's directed by Tom Vogt, and which has a variety of different nano thrust areas, if you will, or focus groups. And so nano-environmental is the newest one. And so we have about 5 or 6 ongoing projects that are really focused on trying to understand the ability of nanoparticles to cause toxicity that may or may not occur from environmental exposures. And what I'm going to focus on are some of the unique or unexpected effects that we've found with a variety of different model systems and particles as well. The first is gold nanoparticles. So there's a lot of literature out there that says gold nanoparticles are considered relatively inert from a biological standpoint. Okay, and that may be true on a larger scale, but on a smaller scale, we are finding that they are somewhat biologically reactive. For example, if you take plants, and these are tobacco seedlings, and you expose them to various size gold nanospheres, 3.5 nanometer and 18 nanometer, you see that the gold exposure to the gold spheres causes necrosis in the leaves. Okay. We also show through a beamline analysis that you can track the gold up through the plant, and it gets up and into the leaves. Okay, but this only happens for the 3.5 nanometer sized particles and not the 18 nanometer sized particles. Okay, so it was a little bit unexpected that we saw such necrosis with these particles, and to try to understand the mechanisms of this, again, we did some more gene profiling studies with tomato plants, looking at exposure to these gold spheres for 5 days. And again, you can look at genes that are upregulated and downregulated and try to get an understanding of how the toxic effect is manifested. And we were really surprised because we expected all these typical stress response genes and pathogenic genes maybe, and genes related to metal exposure, but we didn't see those at all. And so we were pretty surprised, first of all, that we saw such toxicity with them, and second of all, that the mechanism of toxicity doesn't really seem common to other contaminants that we've looked at.